Good afternoon, respected chairman, co-chairman, rapporteurs, distinguished delegates from various institutes. Today I am here to present on the topic exploring the host plant resistance and biochemical responses in Assam rice collection genotypes against the rice leaf folder, that is Cnaphalocrocis medinalis. As all of you know that rice is one of the most important staple food crop of more than half of the world population, and this crop is susceptible to various abiotic and biotic stresses. If you see the biotic stresses, this crop is affected by more than 20 insect pests. They are causing serious damage. Out of the 20 insect pests, 5 are causing national and 5 are having national and international significance. Those are stem borer, plant hoppers, gall-meas, leaf folder, and remaining all causes 25% damage. If we see the paddy leaf folder alone, it causes 10 to 80% yield loss. And this pest is before 1980s, it was a minor pest, but after 1980s, it has started assuming the major pest status due to the use of high yielding varieties and modern cultivation practices. All these are one of the reasons. So still now we are relying on chemical insecticide for controlling this pest. These are some of the negative effects which are associated with the chemical insecticide, like residual effects, impacts on non-target organisms, and there is a development of resurgence and resistance. And this pest, if we see the resurgence, it has developed resurgence for especially for neonicotinoid insecticide like acetamiprid, imidacloprid, thiamethoxam. At present we are using this pest, already developed resurgence for them, and it is also showing resistance to certain insecticides like carbofuran and phorate granules. So in order to rationalize the pesticide use, host plant resistance is the best measure we can follow. So because this host plant resistance is economical, ecologically stable, and we can, it will be compatible with other integrated pest management tools. So for this study, we have chosen Assam rice collection genotypes. Because why we have chosen these genotypes? These are having high biodiversity and wider adaptability. They can adapt at many places of India, and their nutritional value also very high. And besides that, they are having cultural and culinary significance. Contains some of the special features like aroma, color, and their resistance to some of the diseases and pests also. So for conducting this study, it is conducted under mainly two objectives, that is screening of these land rices under the field condition, and next one is analysis of the morphobiochemical traits of these varieties against rice leaf folder. So this was the methodology what we have followed. Total number of genotypes for this study we have used 30 by using the susceptible as TN1. TKM6 was the resistant check for this study. So after transplanting of these genotypes in the field level, we have covered the genotypes after 35 days of transplanting with a green net. We have chosen green net because it allows the 50% of sunlight inside. Almost for 1 week we have covered the genotypes. During that period we released mated leaf folder females, like 50 pairs of adults we have released inside the net. Our main motto of this releasing is which genotype will be preferred by them. It means our basic screening is happening here. So after releasing of the mated females, after 1 week we will remove the net and we will do the scoring by using these formulas based on the number of leaves per hill damaged by this pest. We will score by using the ICAR-NRRI scoring. 2000 recent scoring we are following. This was the actual field layout what we have followed. after every 10 test genotypes, we are planting one resistant check and one susceptible check. And borders are planted with mainly susceptible checks. 2 rows we have used so that it is what kind of selection pressure it is giving, like that we can easily differentiate between the checks and our varieties. So these are the different genotypes we used, and based on the ADLR scoring, that is adjustable damaged leaf rating scoring, we found that these genotypes are coming under 3 different categories. Those are resistant, moderately resistant and susceptible category. Among the resistant category, we have seen 7 genotypes, moderately resistant 11 are falling, and remaining all are coming under susceptible category. So on these varieties, we have studied certain morphological characters like plant height, leaf length, leaf width, and trichome length and density also we have studied. For studying the trichome length and density, we have used scanning electron microscope. So these are our morphological parameters. We found that plant height, leaf length are which are negatively correlated with leaf folder damage, whereas leaf width which is associated positively with leaf folder damage. So here you can see the trichome length and density. These are some of the scanning electron microscope pictures. So we found mainly 2 different kind of trichomes. Those are glandular and non-glandular. Non-glandular trichomes are macro, micro, papillae type and prickle type of trichomes. These are non-glandular trichomes, means they are ductless. Glandular trichomes, they are associated with a duct. So these non-glandular trichomes, how they will protect the plant? So what they do, they will act as a physical barrier so that leaf folder adult cannot come and lay eggs on the particular genotype. So these are all acting as physical barriers. Whereas glandular genotypes, these are associated with a duct, so that they are associated with the releasing of volatile, so that pest attraction towards the genotypes will be reduced. So in our genotypes, we found that in all the categories we have seen more or less both glandular and non-glandular trichomes, but in resistant category, the density and length of trichomes were more when compared to moderately resistant and susceptible categories. Here among these 30 genotypes, from all the genotypes, we have chosen nearly 10 genotypes for studying the biochemical parameters. So we have studied the biochemical parameters like total soluble sugar, soluble total phenol, soluble protein, peroxidase, polyphenol oxidase and catalase we have studied. For this we have used one control and 3 others are as replicates we have taken. So this was the data. We found that antioxidant enzymes which are negatively correlated with leaf folder damage, whereas the total soluble sugar and protein which are associated positively with the leaf folder damage. So these are the certain antioxidant enzymes like peroxidase, polyphenol oxidase, catalase and phenol. This data we have studied both on uninsected and insected genotypes. Uninsected is our control, insected is herbivore pressure induced plants. So that what we found is after when herbivore attack is there, there is a increasing in the production of the enzymes like peroxidase, catalase and polyphenol oxidase we have noticed, and in general also their level of increase is more in case of resistant genotypes when compared to susceptible check. Similarly, we have noticed with the secondary metabolite phenol also. And the other 2 biochemical factors like protein and total soluble sugar, their level is in generality is more in the susceptible genotypes, whereas after the induction of herbivory we have seen that their level of increase is high in case of susceptible plants when compared to resistant plants. So now coming to the conclusions, we found that trichome density is highly, we have seen in resistant genotypes when compared to the susceptible ones, whereas sugar and protein, those are high in case of susceptible genotypes, and the other antioxidant enzymes, those are stress busters. The level of enzyme activity we have seen more in case of resistant lines, and the total rate of increase also we have seen more in case of resistant lines. These are the future prospects, and these morphological parameters and tricho parameters, we found the data resistant lines. Those are helpful for the development of leaf folder resistant varieties, and these lines can be incorporated in the breeding programs and later on helpful for the development of effective pest management strategies, and further we can extend our study for knowing the genomic loci, that is quantitative trait loci, which is responsible for the leaf folder resistance, we will study in the future. These are the things. Thank you.
Thank you so much, ma'am. Okay, there's a small request to the people who have already finished their presentation in this hall, or in fact sitting idle and listening to the other presentation. Some of you, can you please join in the second hall, Subarnarekha hall? The second hall. Well, simultaneously the other presentation are running down. Some of you are requested who have finished their presentation, or their presentation is not lined up in this hall. Please join us in the second hall. We want some audience over there as well. Now, Dr. Arlene C. Alre, ma'am, can we please have you on?